Hello, I'm Max Rempel. My email is here, and I will talk about a model and genomic evidence of imprinting DNA sequence on water structure around nucleosomes. And here is my website with all the publications in full text on the site. I will start with a bit of history. My collaborator and co-author on this work, Dr. Richard Alan Miller, proposed that DNA hologram is the doorway of consciousness. In 1973, he wrote, "We believe that DNA is a projector of the biohologram, both at the cellular level and at the whole organismic level. This means that DNA creates situation of complex pattern of three-dimensional electromagnetic standing and moving wave fronts in the space that the organism occupies. We believe that these wave fronts interact with and interpenetrate with and interdetermine the physical substance that makes up the creature. That's embryonic holography." And he also said in 1973, "Consciousness may be seen as a frame of electrical charges in motion, such as electrons bombarding the television screen. Personality is a time series of these scintillating frames of consciousness. It's a holographic concept of reality." And this is just the nicest illustration of the self-organizing biohologram that I could find. And there is that simplified scheme of DNA bidirectionally connected to the idea of the biohologram, and this one is bidirectionally connected to the idea of consciousness. I will start with a bit of history. In 2019, we published a paper where we connected the DNA and microtubules. So the paradox is that DNA is contained in a nucleus inside, and it is a huge program responsible for the program of the cell, its ability to think, make decisions, pretty complex decisions, regulate large number of biochemical processes and external reactions. And the microtubules, which are outside of the cell, and this is a sensory neuron, the information goes through axon inwards, and in motor neuron, the information goes outwards through action potential. In addition, it was proposed by others that microtubules here are optical fibers, and they're passing optical or other electromagnetic information from the nucleus to the outside to synapses, from the center of the cell to synapses to outside of the cell. So we proposed that there is an interface, electromagnetic interface, between the DNA inside the nucleus through the membrane and the external microtubules, external to the nucleus microtubules. Because if you look at the microscope, the microtubules actually go around the nucleus, and it looks like they are collecting the electromagnetic information, vibrational information from the nucleus, and there is an exchange of information between microtubules and the DNA inside. In 2022, we published a paper, "How Schrodinger's Mice Weave Consciousness," where we propose that collapse of uncertainty in DNA basis is the basis of consciousness and is the quantum molecular basis for the illusion of time. So here we noticed and presented a model that we have in each DNA step, in each base pair, we have a continuous collapse of uncertainty, which is on both electron level. Electrons can be an aromatic state and two non-aromatic states within the base pair. So this is a base pair in DNA. And this is represented by Schrodinger's mice, like 6 mice, which can be either in uncertain state of aromatic or in two certain states of non-aromatic, so non-aromatic states. And also there is an uncertainty of proton positions. There are 3 protons here. In classic hydrogen bond, there are 3 protons. And these 3 protons are oscillating between multiple positions. And that is a known fact. It's a known molecular structure already published and accepted in mainstream literature. So there are 3 combinations, three forms of proton positions. So electrons oscillate, but when protons move in, they stop the oscillations of electrons, stop the aromaticity and rotation of the electrons, aromatic rings. And the observer, and that's represented, the protons are represented by a cat. And then the human observer can observe 3 positions of these protons. And when we are not looking, they are delocalized as well. The electrons are delocalized and protons are delocalized. And at certain situations, this delocalization is collapsed into certainty. So this oscillation between 3 different forms, between the 3 different tautomeric forms of the base pair, happen with about gigahertz or higher than gigahertz frequency. That is a known fact. So, and each our cell contains 3 billions, really 6 billions of base pairs. So we have in each cell, each second, we have a large number of oscillations between delocalized and localized states of electrons and protons. And we suggest that this continuous delocalization and collapse of the wave function is the basis of the illusion of time. Our previous studies focused on oscillations within the DNA, and this is a base pair. And while modeling that, we realized that DNA is bound to the surrounding water by hydrogen bonds. Surrounding water, hydrogen bonds. It is well known that microtubules can propagate their structure, and the snowflakes can also propagate their structure in 6 directions. So we stumbled upon the old idea of others that DNA can also imprint its own structure on water, and we called it snowflake signaling. So the idea is that in a cell nucleus, DNA continuously creates and dissolves water clusters. So now let's bring the field into the picture. In addition to snowflake signaling, which is structural propagation of information, DNA also creates a field. And this field also affects surrounding nucleoplasm, especially when it is in dynamic equilibrium. And it also affects the structure, formation, and dissolution. And it's also a bidirectional process. So the honeycomb hexagonal structure of water crystals is well known. So we also notice that DNA base pairs have hexagonal structures. There are hexagons in each base pair, and there are hexagons between the base pairs. It is difficult to imprint any structure on classical models of ice because they're perfect in each direction and they fit each other only in one way. On the other hand, the flat layers of polywater proposed by Lippincott in '69, they allow and actually require shifts in 3 directions, allowing to imprint the structure on them, and also they have gaps filled with liquid water, allowing for continuous dynamic self-reorganization. Here we embedded the base pair into water honeycomb while keeping the proportions. Here we embedded a base pair into a Lippincott polywater structure. This is a stick model, and this one is ball model, the same structure. Here is a double helix, and the water layers perpendicular to the axis. So how can DNA sequence imprint on water structure? We notice that position of purines depend on DNA sequence, and they can be either on one strand or another. And depending on which position is the purine, the gap between the bases shifts one way or another. And this way it can imprint on water structure. So here you see the base, the gap between the bases is on the left when the purine is on the right, and when the purine is on the left, the gap is on the right. So there is a shift one way or another. So here is the same structure of the shift, and the structure on the left fits brown honeycomb, and the structure on the bottom fits green honeycomb. So the layers of honeycombs are shifted relative to each other. And the shift can happen in any of the three directions. So this way, the DNA sequence would cause shifts in water layers corresponding to purine structure. And if there are two identical DNA sequences, they would fit nicely in this water structure. So where can we find parallel double helix strands? This would be nucleosomes. In our cells, DNA is wrapped around nucleosomes. And nucleosomes look like a magnetic head of video recorder, and it has two parallel double helix strands. The two loops of double helix in a nucleosome are parallel to each other. So if two pieces are parallel on a nucleosome, and if they're connected by parallel water layers, then they might have evolved to have identical purine sequences. And if you unwrap the nucleosome into a string, these red pieces would form vectors. And our hypothesis is that the genome will be enriched in those, we call them jumps. If one vector is repeated again with a gap, and based on the size of nucleosome, this gap would be 77 bases. So we hypothesize that the genome will be enriched with these structures. So we took the whole human genome and selected from it only the parts which are most likely to be participating in snowflake signaling. So we excluded protein coding exons because they're under evolutionary pressure to produce functional proteins. We excluded introns which are transcribed into RNA and are also under evolutionary pressure. And we included only intergenic regions, about 46% of the genome, which are not under evolutionary pressure to produce RNA or proteins. And then among intergenic regions, we only took the part which is bound by nucleosomes, and only the part which is conserved in evolution, meaning that it has function. So we were looking for jumps in a purine sequence, purine jumps, and counted how many jumps we found. And we found that there is a peak with a size around 77 bases in three independent subsets of the genome. And also we found some other peaks which correspond to more turns of the DNA, larger turns of the DNA and smaller turns of the DNA. So this is summarized: 77, minus one turn, minus another turn, plus one turn, plus another turn. So this is a supporting evidence of purine jumps. Therefore, we observed an evolutionary imprinting of water structure on the genome. And there was a second piece of evidence. When we started looking at the turns of the DNA bases, so every DNA base pair is turned around for a fraction of the turn, which is 1 divided by 10.5. Each turn of the double helix contains 10.5 steps of the DNA ladder. And also, we compared it to the internal symmetry of the honeycomb, which has 1/6 of a turn. So the honeycomb repeats itself and you turn it 1/6 of a turn. So looking at those structures and modeling them, we realized that homology jumps will be more pronounced in even nucleotides. So in the DNA ladder, one step would miss the structure, it would be poorly positioned in a honeycomb structure. The next step would be perfectly positioned. The next step would be poorly positioned. The next step would be perfectly positioned, and so on. So then we expect only homology in even nucleotides in a sequence and no homology or reduced homology in odd nucleotides in a sequence. And that is also testable. So the prediction was that in even nucleotides there will be more homology, and in odd nucleotides there will be less homology. So we plotted the same plot and we discovered that it is true that even nucleotides are much more homologous and odd nucleotides are much less homologous in purine jumps. And that's the second evidence that there is an evolutionary imprinting of water structure on the genome. Therefore, this model with parallel layers of water perpendicular to DNA axis is now supported with genomic evidence. So here is the same model. We show one layer of water and two steps of the base pairs. Here are just layers of water in the canyon model where you have gaps which are continuously self-reorganizing. And here is an example from nature. You have a canyon filled with water, and depending on temperature oscillations, the ice would self-organize and reorganize. So our model of continuous self-reorganization of water is directly related to biofield, because the biofield would be most efficient when it would affect the self-structuring of water clusters and continuous self-reorganization. And that's where the consciousness would enter the body in this dynamic self-organizing process. I would like to thank my current coworkers and collaborators and the ones which contributed over a number of years. And here is the website where you can find all our full text publications. Thank you very much.